Welcome back to The Explainer. Today, we're jumping into two just absolutely fundamental processes in all of biology, mitosis and meiosis. You can think of them as life's twin engines, right? But they do very different jobs.
So let's just start with a huge question. Think about it. How do you go from a single cell, just one, to a complex being made of trillions of cells? You. And on top of that, how does life create all the incredible diversity we see all around us?
Well, it all comes down to two different master plans for how cells divide. They're related, for sure, but each one has a super specific goal. And believe me, getting these two mixed up, yeah, that can have some pretty massive consequences.
Okay, but before we can really get into the nitty-gritty of how they work, we've got to meet the key players. It's kind of like you have to learn the alphabet before you can even think about reading the instruction manual, right?
All right, so most of the time, your DNA is just hanging out as this stuff called chromatin. The best way to picture it, think of a giant, tangled up ball of yarn. It's all loose and accessible, which is perfect when the cell needs to, read the instructions on it.
But then, when it's go time, when the cell needs to divide, that messy ball of yarn coils up super, super tight. It packs itself into these dense little structures we all recognize as chromosomes. Why? Well, it's all about making the DNA portable. It's way easier to move it around without it getting all tangled up and broken. Okay, now here's a super important detail you gotta get. After the DNA gets copied, each one of those chromosomes is actually made of two identical copies. We call them sister chromatids. Think of them as perfect clones of each other, stuck together in the middle, just waiting to be pulled apart.
So now we know the players: chromatin, chromosomes, sister chromatids. Got it? So let's look at what these two seemingly different processes, what they actually have in common. What's in their universal toolkit for division?
Turns out they share a lot of the same basic moves. Both of them start by copying all the DNA, then they condense it down into those chromosomes we just talked about. Then they build this kind of scaffolding made of protein fibers called the spindle to line everything up perfectly. And at the very end, they both finish with something called cytokinesis, which is just the fancy word for the cell actually splitting in two. It's this shared toolkit that makes sure the new cells get all the genetic info they're supposed to.
Okay, so that's the common ground. Now, let's get to the good stuff. This is where things get really interesting. This is where those two master plans go their separate ways to do completely different things.
This table lays it all out, and it's actually based right on what you'd see in AP Bio scoring guidelines, so it's super important. Look at mitosis, it's just one division. Boom, done. You get two new cells that are genetically identical to the parent, perfect for growing or healing a cut. But then you have meiosis. That's a whole different ballgame. It goes through 2 rounds of division, and what do you get? 4 cells, and they're all genetically different. These are your sex cells, your sperm and eggs. And see that bit about crossing over? That only happens in meiosis. It's where chromosomes literally swap pieces of DNA. That's the secret sauce. It's what creates all the genetic variation that allows species to adapt and evolve.
And look, understanding this stuff isn't just about passing a biology test. It's about thinking critically. Check this out. This is a real answer from an actual AP exam. A student's score literally depended on how well they could explain these differences. So yeah, this isn't just some random trivia. This is absolutely foundational stuff.
Okay, so think about this for a second. There are billions of cell divisions happening inside your body right now. With that much activity and with so much at stake, you have to wonder, how on earth does the cell keep things from going catastrophically wrong?
Well, it definitely doesn't just cross its fingers and hope for the best. The cell has this incredibly sophisticated surveillance system. It's got these internal checkpoints that are basically like traffic lights, stop and go signals that make absolutely sure one step is finished perfectly before the next one can even begin. So let's walk through these little quality control stations. First up is the G1 checkpoint. This is the big one. It asks, are conditions even right for us to divide? Do we have enough nutrients? Is the cell big enough? Then there's the G2 checkpoint. This is like the final pre-flight check. It asks, okay, is all the DNA copied and is it copied correctly? And finally, you have the M checkpoint. This is the point of no return. Its job is to make sure every single chromosome is attached to that spindle before the cell tries to pull them apart. Each one of these is just a critical gatekeeper.
So what's the engine that actually pushes the cell through these checkpoints? It comes down to this really elegant dance between 2 types of proteins. You've got cyclins. Think of them as the managers. Their numbers go up and down, signaling what phase of the cycle it is. Then you have their partners, the CDKs. These are the workers. They're always around, but they don't do anything until a cyclin comes along and activates them. This cyclin CDK pair, that's the engine that drives the whole process forward.
So, what happens when this incredibly intricate control system breaks down? Well, as you can probably guess, when things go wrong here, the consequences can be really, really serious.
Yeah, when that control is lost, things can basically go one of two ways. If a cell just blows past all the checkpoints and starts dividing like crazy, well, that's cancer. But there's another possibility. If the system detects damage that's just too severe to repair, it can trigger the cell's own self-destruct sequence. It's a process called apoptosis.
And I want to be clear, apoptosis isn't like a messy explosion. It's the complete opposite. It's programmed. It's orderly. The best way to think about it is like a controlled demolition. The cell carefully takes itself apart, piece by piece, to make sure it doesn't harm its neighbors. It's basically the ultimate safety net to get rid of cells that could become a problem. So all of this leaves us with a pretty big final thought to chew on. You have mitosis, which is all about continuity, about cloning cells to keep an individual organism going. And then you have meiosis, which is all about diversity, shuffling the genetic deck to help the entire species adapt and survive. One plan is for the now, for the individual. The other is for the future, for the species. So I'll leave you with this. Which of these two master plans do you think is ultimately more important for the long, long story of life on Earth?